The processes of mitosis and meiosis.
The cell cycle consists of two main phases, interphase and the mitotic phase. Interphase consists of the subphases G1, S, and G2 and accounts for 90% of the cell cycle. The G1 and G2 phases are gap phases in which the cell grows to maturity. In the S phase, the chromosomes of the cell are duplicated in the preparation for division. After the mitotic phase, which consists of both mitosis and cytokinesis, the daughter cells repeat the process and the cycle continues.
Mitosis, cell division.
Mitosis is broken down into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. G2 is the final stage of interphase and occurs before mitosis. During prophase, chromatin fibers condense into chromosomes that appear as identical sister chromatids joined at their centromeres. The nucleolus disappears, and mitotic spindle forms separating the chromosome. During prometaphase, the nuclear envelope fragments. Specialized proteins called kinetochores form at the centromere of each chromatid. Microtubules extend from the centrosomes through the nuclear area and attach to chromosomes at their kinetochores, becoming kinetochore microtubules, and non-kinetochore microtubules connect to those coming from the opposite pole. During metaphase, the chromosomes line up their centromeres at the metaphase plate, a plane at the center of a dividing cell, and each sister chromatid is connected at their kinetochore to kinetochore microtubules that extend from centromeres at opposite poles of the cell. During anaphase, the microtubules separate the sister chromatids, forming identical chromosomes that are pulled to the centromeres at opposite poles by shortening kinetochore microtubules as non-kinetochore microtubules lengthen and elongate the cell. During telophase, the fragments of the parent cell's nuclear envelope and chromosomes form two identical daughter nuclei in the cell. The nucleoli reappear as chromosomes become less condensed and the spindle disappears. The process of mitosis is now complete. Cytokinesis occurs at the end of telophase and involves the formation of a cleavage furrow that pinches the cell into two daughter cells that contain the same number of chromosomes as the parent cell.
Meiosis 1, separation of homologous chromosomes.
During prophase 1, chromosomes condense, homologs pair up and align along genes. Crossing over, the genetic rearrangement of corresponding segments of DNA molecules in non-sister chromatids occurs as the homologs are joined in synapsis. After synapsis, each homologous pair has chiasmata, X-shaped regions at the point of crossing over. Other processes that occur in mitosis, such as centrosome movement, spindle formation, and nuclear breakdown, also occur. During metaphase 1, homologous chromosome pairs line up at the metaphase plate and kinetochore microtubules from one pole attach to both chromatids of one homolog, while kinetochore microtubules from the opposite pole attach to the other homolog. During anaphase 1, the proteins that hold homologs together along sister chromatids are broken down to allow the homologs to separate and move to opposite poles while sister chromatids stay connected. During telophase 1 and cytokinesis, two haploid cells that each consist of a complete set of duplicated chromosomes composed of sister chromatids containing cross-chromatid DNA are formed by cell division.
Meiosis 2, separation of sister chromatids.
During prophase 2, spindle apparatus forms and any part of the nuclear envelope that forms is degraded. During metaphase 2, chromosomes that are not identical due to crossing over in metaphase 1 line up on the metaphase plate and sister chromatids are attached to kinetochore microtubules extending from opposite poles. During anaphase 2, sister chromatids separate and are pulled to opposite poles by kinetochore microtubules. During telophase 2 and cytokinesis, 4 full haploid cells are formed from the original parent cell that are genetically distinct from one another and from the parent cell, and contain a haploid set of chromosomes. The complete cycle of meiosis. The main difference between mitosis and meiosis is that mitosis forms two identical daughter cells that contain the same number of chromosomes as the parent cell, while meiosis forms four haploid cells that contain half the number of chromosomes as the parent cell.